We've covered a lot of ground in this chapter looking at cell reproduction, and hopefully some of these words look pretty familiar to you. So looking at them very quickly: interphase, prophase, prometaphase. We're seeing the different stages of a life of a cell, and then we go in, here's interphase again, and then we see the G0, G1, S, and 2. Talk about mitosis and the mitotic phase. Some of the lingo that surrounds the actual cell, particularly in mitosis, when we're starting to look at how mitosis goes through these stages. So we see centriole, mitotic spindle, kinetochore, etc. But now we're going to see how all this is regulated. And we know that the cell cycle, we can see it right here, and we can see that here's the interphase is this gray all the way around, and that this in the purple we see that this is the stage, this is the G1 phase of a cell's life. So it's been born, and now it's growing up, and it's grown up, and does whatever its job is. So a muscle cell is doing muscle cell things during this G1 phase. But at some point, the cell could receive some signals, and those signals can come internally or from the outside. So those signals we're going to talk about here in a minute, and the cell will get that signal to say, let's move forward. We need to go through cell division. We're going to make another copy of the cell. But it won't go through without passing by these things called checkpoints. So we have a checkpoint here at G1. Your book, in the diagram in your book, it says there's a checkpoint at G1, at G2, and at the M phase. But there's also, and it explains a little bit on this, there's also a checkpoint in the S phase when the DNA gets synthesized. And in truth, there's probably several checkpoints that we're not going to talk about here because it's really can be a complex process. But the take-home message is the cell is trying to ensure, or our body has the genetic code to ensure that we're not going to pass on information to another generation of cells if it hasn't been thoroughly checked and be sure that it's all in the best possible shape. So we're going to talk about each of these checkpoints in turn. I added this into our slide set, so you'll notice that this is a little different than the slides I shared with you. So we're going to see a couple of different regulatory things that regulate this cell cycle. Now, what are some of those things we're talking about that even tell the cell what to do? Well, there are external triggers that can either start the process of the cell cycle or inhibit the cell cycle. And you can see what they're listed here, right? So if we have a death of nearby cells, some of the surrounding cells can send out messages to some of the healthy cells in that perimeter and create the process or start that process of cell division. There could be growth hormones that come in because this individual is growing, or some cells need to increase the size of a particular body part, an organ, etc. So that's the release of growth hormones are going to promote that. Cell crowding can stop the process of the cell moving on in the cell cycle because that's a signal that we don't want too many cells, right? So we're going to control that too. So all these are external triggers. So there's also internal factors that are going to regulate this process. So let's talk about a few of them here. Starting with G1, that checkpoint is going to determine whether or not all the conditions are right for a cell to go on and move into ultimately mitosis. So always the question is, do we have enough of everything on board in order to go forward? Do we have enough energy, etc.? So is the cell of a proper size? We don't want the cell to, once it's been born, it has to grow up a little bit, so we want the cell sizes to be correct. We also want to check to see there's no damage to that DNA. So that cell could be a cell that's been around a while, and maybe you could even think about it as a skin cell A skin cell receives a lot of ultraviolet light. We want to be sure that skin cells' DNA hasn't been damaged before copying all that and potentially sending on damaged DNA to the next generation. So we'll talk about one of the regulatory molecules coming up called p53. If a cell doesn't meet all of these requirements, it's not going to move into the S phase. As a matter of fact, it might be put on a temporary hold into the G0 phase until all of this is determined. We're going to see how that works. So here's that G1 right here. So here's G1. It's when the cell is doing its job and then ultimately gets some signal that says you need to make a new copy. So if all those things aren't correct, then the cell is not going to let that cell move past this checkpoint right here. And I'm going to talk about those specific regulatory molecules too in a minute. So if there's a problem, then there's that molecule, there are a couple of molecules. One, I'll talk about this one too, something called P21. We'll get into detail again on this a little bit later. So if the cell gets a signal to go forward, but there's a problem, there are a couple of things which can halt that cycle from moving past this checkpoint and can send that cell into this period we're calling G0 to wait until we're sure that cell is ready to move on. We've checked the DNA, corrected any errors if we can, fixed the damage, etc. Now, just as a side note, G0, some cells are always in G0. So cells that don't go through mitosis, that don't replace themselves, are going to go into G0 and be there their entire existence. So a lot of our neurons, our nervous system cells, are born, grow up, finish their development, and then go into G0. So they still do their job. They just aren't ever going to go, that they aren't going into the cycle such that they're going to go around the cycle again and make a copy. The S phase, so the synthesis phase, so let's say we've passed G1, we go into the S phase. The S checkpoint, which I wrote this slide in, but is not shown here, so you don't see any little box that's showing a checkpoint. But again, we got to check to be sure everything's correct. So DNA has been properly synthesized here. We've copied all of that DNA. We've replicated, another term for all of synthesis, replicated that DNA. No damage, everything's good. So we want to be sure of that before we go into the next phase, which is G2. So G2 is like the last ditch effort to be sure everything is correct and to be sure the cell has everything it needs to move forward. Its size isn't sufficient, it has enough proteins in there, it has enough ATP for energy, and all of that DNA is in good shape. So here you see it in the cycle again. Again, here's P53. So remember, this is our DNA damage checker. And so we'll have P53 involved here. If something is detected at this point, something's wrong, maybe it's escaped some of these other detectors, but we're seeing something that's not correct, then again, the cell is going to stop from moving forward. It won't go into mitosis at first to determine whether or not the damage can be repaired. Now there's a term, if it can't be repaired and the cell determines that that cell needs to go away and not go through mitosis, it can enter a programmed type of cell destruction called apoptosis. We're going to see that again. So let's say everything's good. Now we've gone into mitosis, and there's another checkpoint at the end of metaphase. And that metaphase checkpoint is going to be sure every one of those sister chromatids is attached properly to the spindle fibers and will not go forward unless that's true. Okay, so the cycle is going to be halted if those spindle fibers aren't properly attached to those little hook-like structures we mentioned before called kinetochores. If that didn't stop that process. If at this point there was an issue, or let's say that the checkpoint failed, then some of those chromosomes, or those sister chromatids, could end up both being pulled in one direction, and then another one of the daughter cells would have too many, and one of the daughter cells would have too few. So here's that here's the M checkpoint. I'm going to talk about this coming up, these two regulators, something called MAD and something called APC/C. So we're going to look at these and what they do to either arrest or stop the process or to push the process forward. So these two regulators are types of regulators exist, and it's kind of a group of them, right? And the positive regulators are ones that are going to move the cell cycle forward. The negative regulators, those which are going to stop the cell cycle from moving forward until it's determined that all is well. This link right here is going to take us to this biointeractive page that you're going to be working on this week. There it is, on the cell cycle. So let me move this out of the way and see if we can get just to the background information right here. Let me go forward. Sorry, it's making me do that. There we go. So here you see it again. Here's the cell cycle, nice and pretty and colorful. And we can go through each one of these parts of the interphase, G1, S, and G2. And when we do that, what we're going to see is the description of what those are. Here's the S, here's the G2, and then we also can do the same for mitosis. So, and within each one of these are checkpoints. So here's mitosis, and I can click on that checkpoint, but really where I'm going to find information mostly about it is to go to this lower part that says cell cycle regulators in cancer. And here you see the positive and the negative regulators, kind of like our green and our red, showing sort of like go and stop. Now, the things which are positive regulators are those which are going to push the cell into the next phase. These positive, so there are genes, though, that encode those particular proteins to cause those things to happen. And the genes that code for the positive regulators are called proto-oncogenes. And the genes that code for the negative regulators, or the inhibitory things, the things which are not going to allow the cell to move forward, are those inhibitory proteins. Those are these called tumor suppressor genes. So that when we say the genes, so within the DNA are genes that are going to code for these various proteins. Those which code for those regulators that move the cell cycle forward are called proto-oncogenes. Those which code for those proteins which will stop the cell cycle from moving forward for one reason or another, those are called tumor suppressor genes. And so that's what's being represented here. Now, the most important regulator in moving this cell cycle forward are something called cyclin-dependent kinases, or CDKs. And cyclin-dependent kinases, remember the word kinase has to do with phosphorylating something. So these are enzymes that are present at the various parts of this cell cycle. And they're going to respond to the making of some substrate. So we're going to see that in a short time. But they're going to, by their activity, they're going to phosphorylate or dephosphorylate something to either activate it or inhibit it. So that's what these are. And you're going to work through these together in class. So I'm going to return to our slides. Now, we said that those positive and negative regulators, and particularly this one I just mentioned, this thing called CDK cyclin. Sorry, I'm trying to fix the screen so there's a little bit more room so you can see more of it. But what we see here is these things called cyclins. And cyclin, there's 4 different flavors of cyclins here, and these proteins are going to be, they're coded by those proto-oncogenes in their DNA. So they're going to be made in response to something activating it and producing these cyclins, and then these cyclins are going to bind to those kinases that I just mentioned. So a positive regulator works like this. What you see here is the cyclin, I'm going to move this up again so you can see it, cyclin-dependent kinases. So here's our CDK, that's our cyclin-dependent kinase. And here's cyclin. So cyclin was produced by the cell in response to some signal. A proto-oncogene was activated, started making the cyclin. So now cyclin is made, and it binds to this enzyme, CDK. And when it does that, it also has this effect of connecting with a phosphate. So that's this little kinase piece of it that we see here. A phosphate donating protein is going to be involved. And then once this all forms, this becomes this complex. And the CDK cyclin complex is formed with this phosphate on it. And now it can go out and use that phosphate to activate or inactivate various other proteins that are around. So this one you're seeing here, the CDK cyclin that is now phosphorylated, is activated. It's moving to some target protein. It's donating that phosphate group onto it, and that donation of the phosphate group is going to, on that target protein, is going to activate the target. So we're going to see how that works. So here I gave you a little bit of text on here to explain what we see as CDK cyclin with that phosphate is going to go phosphorylate one of the major inhibitors of the cell cycle at G1, something called RB. And it's going to inhibit the inhibitor, basically allowing the cell to move forward under the good conditions. So we have to talk about that a little bit. The negative regulatory molecules, the ones that would be under that tumor suppressor gene category, are ones that the first one I just mentioned, RB, and then back to this P53 and another one called P21. They all act at somewhere around that G1 checkpoint to control the activity so that the cell doesn't move in to the cell cycle unless necessary. Now, this picture here I'll explain more in class. What we see here, we're looking at is the DNA. So we're looking at some gene on this part of the DNA of the cell that is able to be what's called a transcription factor. So I don't want to get into all of that here because we're going to talk about that coming up. The final thing is looking at cancer. So what happens when one of these regulators isn't working properly? So we'll talk about this also. I'm going to do this a little separately because what I want you to do first is to go through that activity where you're learning about the cell cycle, those cell cycle regulators, and then we'll go from there. Okay? Alrighty.